Okay, who's got questions? Yes.
Can you talk a little bit about like the facial projection and how that changed between like Homo erectus, Homo habilis, and like Homo sapiens?
Okay, so facial projection is generally just an index of how far the facial features stick out from the center of the skull. And what do you suppose we used as a baseline for that? What species had a facial projection index of 1? What? Anybody? Not start with a P. So what species of hominid has a facial projection index of 1? Or in other words, what is the baseline we use for our facial projection index when studying hominids? Did anyone study this before they came in? These review sessions are generally meant to be after you've done some studying. If you were going to make a facial projection index, what would you use as the baseline? There you go. So we use ourselves as the baseline for the facial projection index because we are self-interested. So when we're comparing our ancestral hominids to ourselves, is that not unlocked? Hang on. So when we're using that facial projection index to compare ourselves to other hominids, we use ourselves as the baseline, and things that are more different from us have an index that deviates more from 1. So generally, as our ancestral hominids are getting closer and closer to us, or more closely related to us, you're going to see the facial projection index looking more and more like it does in a human. Does that answer your question at all?
Yeah, when it gets like further away from us, does it get bigger, smaller, or just like different, like either way?
I don't know for sure on this one, but I think in all cases it should get larger when we're talking about ancestral hominids.
Okay.
Because if you look at the skulls of the ancestral hominids, so let's say we're taking H. neanderthalensis and comparing it to humans. If you're looking at the projected facial features like brows and such, you'll notice that they stick out much farther. If we go farther back, they get even more projected and even sticking out even more than humans, which would generally result in a facial projection index larger than 1. All right, does anyone have any more questions on that specific topic? Generally, anything I ask or might ask about facial projection indices on the exam would be more in the general sense. Like, I'm not going to say, what is the facial projection index on Neanderthals? That would not be a really reasonable question. All right, what else we got? Yes?
What's the difference between when we use a one-tailed side test and a two-tailed side test?
So, do you remember what we use the sign test for?
Yes.
What is it?
For p-value.
Well, p-value is what you use any statistical test for. What specifically are you using a sign test to analyze?
Like,
What's that?
Like statistical significance.
What's that?
Statistical significance, like between two.
I mean, that comes back to p-value, but you're trying to compare, basically, generally you're going to use it for count data. So you're going to be comparing counts of certain events from two different groups. So do you remember the difference between a one-tail and two-tail sign test?
Was one-tailed when you had like no prior knowledge and like
So one-tail is really if you're only interested in testing whether one is higher than the other or whether one is lower than the other, but not both. In a two-tailed sign test, you're looking to see whether it is higher or lower. That's why it's two-tailed. So a one-tailed sign test you would use if you're only trying to determine whether one group has a significantly larger count than the other, or whether it has a significantly lower count than the other, but not both. You're not looking at both. Like you know that one is going to be higher than the other, you're just trying to see if it's significantly higher. With a two-tailed sign test, you're looking to see if it's significantly different in either direction, higher or lower. Like an example for that might be if we're trying to compare the facial projection index for Neanderthals and humans. We expect it to be higher, but we're going to see is it significantly higher. That would be a one-tailed sign test you use for that. If it were something else like, I don't know, if I want to see if the students in my afternoon lab are taller than the ones in my evening lab or shorter than the ones in my evening lab, then you could use a two-tailed sign test. Because I'm not sure if it's going to be higher or lower. Does that make sense? Great. Does everybody know what a p-value is? A lot of you struggled with that one last week. Who can define p-value? You get a p-value for any statistical test you do, generally. And what does that p-value tell you?
like whether the preference is significant or not?
No.
Okay, good.
Literally, what does the p-value tell you? Who can define p-value? You guys forgot this already? You had to do it last week.
Isn't it like the chance of like rejecting the null when it should not have been rejected? Or like the
So, yes, but a slightly better way, a slightly more descriptive way to say it would be it is the probability that any differences you see in your groups are due to random chance. So it's important to keep in mind what a p-value actually is. Most people know more or less how to interpret it, because if the p-value is greater than 0.05, you say these results are not statistically significant. If it's less than 0.05, you would generally say they are statistically significant. But what that actually means is you've set the cutoff at a 5% probability that any differences you see between your two groups are just due to random chance. If it's less than 5%, then you can confidently say these are probably different groups, significantly different. Does that make sense? So if you have a p-value of 0.34, we'll say, that means there's a 34% chance that any differences you observe are just due to random chance. And that's a high enough probability that you wouldn't say with confidence, yes, these are two statistically significantly different groups. Everybody got that?
Do you have to know the formula for the p-value?
No. You guys won't have to have any formulas memorized, but any formulas you've seen throughout the last half of this semester, you should be able to recognize if I give them to you, and you should know what each variable in the formula represents. Yes?
Can you talk about what standard error means? Because I know it's like the denominator.
So standard error is generally, it's just a measure of deviation from the center, whatever measure of center you're using, usually average. There's a bunch of different ways to, or there's a bunch of different metrics of variance from center that people use, one of which is actually literally called variance. There's also standard deviation, standard error. I won't make you calculate standard error unless I give you the formula for it. And the most you would have to know in general if I asked you what it explains is that's giving you a measure of how far from the average your values are. Generally, in most cases, when you're reporting data, it's pretty worthless to just give an average. You never give a measure of center without some measure of spread, either standard error, standard deviation, etc. Let me see if anybody emailed me any questions. That might make it easier here. It looks like no one emailed me any questions. All right. That's for a different class. Yes.
For the tide pools, when you ID the basic animals, like to what extent?
The most specific I would want you to be able to do is if I give you a picture of a chitin, tell me it's a chitin. If I give you a picture of a barnacle, be able to tell me it's a barnacle. You definitely don't have to give me the binomial names. You should also be able to tell me what phyla they may belong to. Like, who can tell me what phylum the barnacles are in? Nobody? Arthropods, yes. How about mussels? They are mollusks, yes. So those are the kind of questions I might ask. You should also be prepared to potentially describe some specific adaptations of specific organisms that they've developed in order to overcome the challenges of living in the intertidal zone. Like the mussels, for instance, they're really good at tolerating air exposure because they can just close up their shells and seal up and keep some water in there and not dry out. And that allows them, for instance, to live higher up in the intertidal zone where some other intertidal organisms can't live. Conveniently, their main predator, the sea star, can't live as high in the intertidal zone as they can because they can't tolerate as much air exposure. So you should have a general concept of the different challenges, the different limiting factors in the intertidal zone, some of the different things organisms have evolved in order to deal with that, and what that means in terms of distribution and life in the intertidal zone. You don't have to know everything, but be prepared to give a couple of examples if I say, you know, what are some specific examples that some organism has developed. And you can choose the organism, you can talk about sea stars, you can talk about barnacles or something like that. Yes?
So the question in that says, how do the factors affect the distribution in the tide pools?
Yeah.
So like what would an example of that be?
Basically, like I just said. So the one factor or one challenge that intertidal organisms face is dealing with air exposure or desiccation as the tide drops. So based on how well those animals can handle air exposure, based on the adaptations they've evolved, that determines how high up they can live in the intertidal zone. So that determines their distribution. Generally, the upper limit of just about any organism is going to be how much air exposure they can tolerate. The lower limit may then be defined by something else. Like the lower limit of distribution of mussels in a healthy intertidal ecosystem is how high up the sea stars can go and eat them. Stuff like that. I may ask you to give a specific example of some specific challenge that all organisms face in the intertidal zone, then give me a specific organism that has a specific adaptation to overcome that challenge. Like, for instance, I might say, well, I shouldn't give away any questions, but you guys get what I'm talking about, right? If I were to be really general, someone name a challenge in the intertidal zone, other than the one I've been talking about this whole time. Yeah, wave energy. Waves are breaking directly onto the intertidal zone a lot of the time. So what's some organism that's developed some adaptation to deal with that? Good thing you guys came to this review session. Okay, sea stars. What's an adaptation they have to deal with that challenge?
They have the
2 feet, which allow them to finish the sentence.
stick to like the ground, to rocks.
Rocks, that's a good way to describe it. Yeah, so that would be a good example. Now do you guys see what I'm getting? Yeah, great. Yes?
With the wolf and like moose thing, why when there's a longer like growing season or like breeding season or whatever, that cause like the margins between the highs and the lows for the wolf and the moose to like increase?
What do mooses eat?
Plants.
would those be affected by a longer growing season? Yeah, so if you have a longer growing season, there's more food for mooses. If there's more food for mooses, are there going to be more or less mooses? Generally right. What do wolves eat? Many things, but also the moose. So if there's more mooses, does it stand to reason there could also be more wolves? So do you now see how the growing season can affect both populations?
Yeah. And like what causes them to go down? Just the opposite of like
What happens at the end of the growing season? Yep. Plants stop growing. What happens then? Is there more or less food for mooses? Less. So are there going to be more or less mooses? Less. And if there's less mooses, is there more or less food for wolves? Generally less, yeah.
I have one more question about
yes.
it too. I was talking about the fat stores of moose, and there were wolves and there weren't, and it's saying when there's more fat stores, there's more wolves. You know, tie that together.
Who can make sense of that? When there's a higher wolf population, you see higher fat stores in the mooses. Go.
Wolves will they'll like eat more mooses so that like there's less moose like eating plants, so then the moose that are alive can eat more plants and get fatter.
Nailed it. If you have a higher concentration of predators, you have a lower concentration of prey. If you have a lower concentration of prey, then there's more food per individual prey to go around. So the individual prey can get fatter. Make sense? Great. All right, what else we got? Yes.
So, it says, what is a wolf's starch in the plant, and how can you tell if the wolf doesn't possess this starch within its body? So the wolf is the stored energy. I don't know.
Yes, you do. You did it in one of the labs. Iodine test. Yes.
Wait, what is color change? Is it like
What color is iodine normal?
purple?
Yep.
Yellow.
Yeah, amberish, I would say. In the presence of starch, it becomes purple. Yes. In high concentrations of starch, it becomes so purple it almost looks black. That's the best way I know of to tell if there's starch in a tissue. You guys have any starch in your tissues? No. What do we have instead? What do we use as long-term energy storage? It's another polysaccharide. No. That is a lipid. We didn't go over this in class, so. What? Glycogen, exactly. So glycogen is another polysaccharide, which is a branch. It's a more branched chain of glucose than starch is, but that's what we use for moderate long-term energy storage in our muscles. That won't be on the test though, because it had nothing to do with any of our labs. Still worth knowing. Yes.
Let's talk about like the hermit crabs, like the mechanistic versus adaptive. Like reasons, for example, of behavior.
So, exactly what parts? Make sure I'm giving you the right answer. can't organize my thoughts clearly enough on that, so let's just say there won't be any questions about how to test.
Love it. Adaptive
So which exact part of that? 1C?
Yeah, once.
All right, ignore part 1C in the study guide. That won't be on the test.
So do we even need to know about mechanistic?
I'm not going to ask you to define or compare contrasts. Yeah, but I can't really organize my thoughts well enough to ask a good question about that. Okay, what else we got? Yes.
Why? Okay, so I understand why like mosses rely on the water environments, but why do the seedless vascular plants still rely on them?
Okay, who can explain this one? Why do seedless vascular plants still rely on wet environments?
It's actually like the gametes, because like even though they still like have vascular tissue, whatever, their gametes still can't start without reliance on water.
How would you describe the gametes of seedless vascular plants?
Not
Particularly the male gametes.
fit to swim.
Yes, they are flagellated sperm. They have to swim to the ovule. So they still require a fairly moist environment for reproduction. At least sexual reproduction. How else do they reproduce?
Asexually or unisexually, right?
Asexually is the right descriptor,
okay.
but more specifically, mechanistically. See, we're already doing it. What is the other thing that seedless vasculars use to reproduce?
Like spores.
Yeah, exactly, spores. What do we call the structures that produce the spores on the fern? And where are they located? Yeah, a sorus singular or sori plural. And they are found on the undersides of the leaves of the gametophyte or sporophyte structure. Yes, sporophyte structure. What does a fern gametophyte look like compared to the sporophyte?
Smaller.
Yeah, much smaller. Yes.
I just want to like check for this one. It asks why sea plants can dwell in a greater range of habitats of bryophytes and ferns. Is that because of the seeds?
A little bit louder.
Like, is that because of the seeds? And that
Is
they can
what because of the seeds?
why the sea plants can dwell in a greater range of habitats than the bryophytes and ferns?
Can what in a greater range?
Can dwell.
Oh, can dwell in a greater
Yeah.
range of habitats. Okay. So seed plants possess a number of different adaptations, primarily regarding reproduction. So what is the advantage of a seed?
It like protects the embryo.
Protects it specifically from what?
Desiccation.
Desiccation. And it also allows it to wait. Yeah. So the little embryo inside that seed has a big bag lunch in there basically. So it can just kind of post up and wait for optimal growing conditions. So that allows seed plants to live in much drier habitats. Their gametes also have adaptations to prevent desiccation. You have those you have the pollen grains that are covered in sporopollenin, which prevents them from drying out. So their gametes can be spread without water, on the wind, or on the legs of a bee. Yes.
In terms of conifers, are the males usually? At first, I thought the males are usually higher because of like the wind distribution.
Yep.
Is that right?
Yeah. So in most cases, when you have monoecious conifers, where you have both the male and female structures on the same plant, the female cones will be found lower on the tree, the male cones will be found higher, and that is generally because they rely on wind to disperse the male gametes. You should also be able to recognize male versus female cones if shown in pictures, or if I bring in actual cones. At this point, though, I plan on only using pictures for the practical.
Is it gonna be the practical gonna be focused on one topic or like multiple?
Everything. Everything we've done is fair game for the practical. Yes.
And on like the practical thing, it says you'll be able to identify whether you're looking at a sporophyte or gametophyte on the practical station. I don't really know how it's supposed to.
So if I give you a picture of some part of a plant, you should be able to tell me if it's sporophyte or gametophyte. How are you going to tell on the higher plants, for instance, the angiosperms? Yes. So what's the gametophyte structure going to look like?
Super small.
Yeah, literally microscopic. Now, if we're talking about some of the lower plants, like Cebus vasculars, for instance, then the gametophyte stage is not necessarily going to be that small. So if I give you two pictures of a fern and say which one's the sporophyte, which one's the gametophyte, you should know.
Wait, can you say it again?
If I were to show you two pictures of a fern at different stages of its, or different generations of its life cycle, you should be able to tell me which is the sporophyte and which is the
Is
gametophyte.
that like the story? Or
Well, you may be able to see sori in the picture, or you may not.
So how would you tell? Like,
Alright. So everybody knows what a fern sporophyte looks like. Yes, you do. So this is the fern sporophyte structure. This is the fern gametophyte structure.
oh,
You should be able to tell me which is which if I show you pictures on a slide. Does that seem fair?
yeah.
yes. How do we tell that for like other
Yeah.
plants?
Yeah, can we just use the fern?
Well, you should be familiar with the sporophyte and gametophyte structures in like liverworts or something, or mosses as well. So,
Yeah.
if you're talking about mosses, they exist at the same time. So here's a moss where you have both at once. I might have arrows pointing to each one and say which one is the sporophyte, which is the gametophyte. Yes?
Would you like say if it's a fern or whatever?
Yep.
Okay.
Well, I might, but not necessarily. I would absolutely expect you to be able to recognize like a fern from a
Right.
moss from a tree. You should definitely also know which ones produce seeds, which ones have vascular tissue, stuff like that. Yeah, definitely be able to tell me whether it's a bryophyte, Cebus vascular, gymnosperm, or angiosperm if I give you basically any plant. They all share enough of the same characteristics that you should be able to tell me, even if you've never seen that specific plant. It should be relatively obvious. I will not pick any one that would be really hard to tell. Yes.
What do you want us to know about the sagittal crest?
So basically, what is it? And sagittal crest. Anybody remember what that is?
Muscle point, like muscle attachment point for jaw muscles.
Sagittal crest, right here. Do you see that in Homo sapiens? No, not really. Yes.
So the foramen magnum, can you just clear up
So foramen magnum is not going to be, oh, you mean, okay, I see what you mean. So that is basically, foramen magnum, hopefully everybody remembers, is this guy right here. This is where the spinal cord and brainstem go into or out of, depending on which way you're looking at it. So you can generally tell what the posture of some organism is going to look like by looking at where the foramen magnum is on the skull. Okay, this one's hard to see, but the foramen magnum in this case is like right here. It's very posteriorly located. If you have a very posteriorly located foramen magnum, what is your posture going to look like? Not very upright, probably, because otherwise your head is always going to be like weighted down. So the positioning of the foramen magnum on the human skull near the center is one of the things that allows us to stand upright, because our head is relatively balanced over our body. So I guess in that case, be prepared to explain briefly how the positioning of the foramen magnum relates to the posture and therefore the lifestyle of the organism. Basically what I just said. Or also, if I were to just give you a picture, you should be able to more or less guess. Although I'm not going to give you a picture of the bottom of the skull and ask you what species it is. Just like, based on the positioning of the foramen magnum, what do you suppose the posture of this organism looks like? And you may need to draw general inferences of what its lifestyle then might be. Or maybe just give examples of some specific benefits of the position. Like the fact that we can walk upright gives us all kinds of benefits, like carrying stuff while we walk, like our babies or something like that. Also makes it easier to use tools. Speaking of bone structure relating to posture, you should also be able to recognize roughly what the posture of an organism might be based on relative lengths of different bones in its body. Like if something has relatively short arm bones and relatively long leg bones, what would you expect? What's the posture going to look like? Pretty upright. If it's the opposite, then maybe less upright, maybe more quadrupedal. If I remember correctly, the section of the lab manual on primates had a fair amount of explanation on this. If not, I know the lecture did. Yeah, I guess it was all in the lecture. So just be generally aware of the stuff in the lecture on that section. Everyone knows those are all on Blackboard, right? And everyone found the study guide on Blackboard? Great. Okay, what else we got? Would you guys be comfortable identifying population growth curves? Logistic growth versus exponential growth. Would you be comfortable identifying carrying capacity on one of those curves? Okay. Is anyone not? Are you guys just nodding because you don't want to be different? Yeah, so like for instance, if I were to give you this, what type of growth is that? Logistic. You can see because it levels off at this asymptote. What is generally going to be denoted at this asymptote where it levels off? What do we call it? Very good. What determines carrying capacity? Yeah, in general, the environment. It could be more one factor or more the other, but things like amount of resources, amount of predators, stuff like that. Population ecology is really cool stuff, and I wish we spent more time with it, but we didn't. Yes.
Can you go over succession?
Which part of this did you get? Okay. So, I haven't actually written the exam yet. I need to go into the Isla Royale activity and see exactly what types of questions I should be asking. But in general, succession is the procession of organisms taking over one after the other. Like after some sort of disaster has happened that wiped out most or all of the organisms, you have succession of this comes first and then this comes next and kind of takes over and then this one takes over, stuff like that. So in the instance of like plant succession or something like that, if everything gets wiped out down to bare rock, then the first things to come in are like mosses and lichens and stuff like that. As they decompose, they start to form soil for slightly larger plants to take root in. Then those plant roots are excreting CO2, making the soil slightly acidic and breaking up the rock underneath it, which allows larger plants, which then shade them out, and then larger plants still come in and shade them out. That's succession. Yes?
So on this diagram, there's nothing about like knowing like simple stuff which we need to know, like kind of specific stuff.
Not super specific stuff. Mainly anything I would be asking on the exam would probably be relating to the ecological relationships between the wolf and moose. Like what we talked about earlier about why do you see higher fat stores in the moose when there are more wolves? Something like that. Or, you know, if something happened to the wolves, what would you expect would happen to the wolf population? If there are more wolves, what happens to the moose? If there are less wolves, what happens to the moose? Stuff like that. Or I might get even more complicated and say if the wolf population increases, what would you expect would happen to the plant population? You should be able to predict that an increase in wolves means a decrease in mooses, which means that there's less mooses grazing, which means there's going to be more plants. Stuff like that. Yes?
One question says, what trends were seen in skull indices over time in the age group? I get what you're talking about facial projection and like about close to Homo sapiens. Other than that, what else should we know about that? I get like not specific numbers, but like
Yeah, there won't be any
in terms of, like,
specific
trends,
numbers.
Like I don't even know the trends.
So it's tough for me to talk about that one since I actually haven't written the exam yet. I doubt I'll go too in depth with that. Like I doubt I'll be asking about, you know, skull proportion indices of prosimians or something like that. That would be unfair.
Okay.
Probably it would be generally similar to what I was explaining about with the facial projection index, where as they get more and more or closer and closer related to humans, you would expect to see indices approaching 1,
Okay.
because we base all of our indices off ourselves because we tend to think we're the most important animals in the world. And also it's just easier for us to conceptualize our own skulls. Everybody knows what a person looks like. Not everybody knows what Australopithecus africanus looks like.
Yeah.
Yes?
Are both hominoids and hominids monophyletic?
What would you guess?
Yeah.
Why?
Because the graphs in the slides, when we box them, they look monophyletic.
So what are the hominoids?
They're after prosimians.
Probably more specific than that. What? Yes, so who can define monophyletic?
Like a common ancestor in all of its descendants.
Right. So what common ancestor would the hominoids share? Well, would they share a common ancestor with humans? Yeah. Then would the humans be included in the hominoids? Or would the hominids be included in the
Yeah.
hominoids? Are they?
They're in the box.
Let me see what slide that was. Do you remember?
I can find it though.
Yeah, if you can find it, that'd be great. And I'll also look for it real quick.
Yeah, it was 16.
Alright, slide 16. Go away.
Because then also the hominids or bipedal hominoids later. But then just on slide 16, the like red box.
Well, PowerPoint's not working. There we go. Alright, you said it was slide 16?
Yeah.
Alright. Oh, apes, or hominoids just as apes. Yes, okay. Yeah, in that case, yes, they would be monophyletic. So in this context, hominoid is just another word for the apes. And then the hominids, I would also say are monophyletic because you have this group. So yeah, to make a really long answer short, yes.
Okay. And then prosimians are paraphyletic.
Yes, and why are they paraphyletic? So on, let's see, on this slide, God, why is PowerPoint not functioning? Let me full screen it. Okay, what are the prosimians? PowerPoint is not going to cooperate. Specifically, list everything that's included in the prosimians according to the slide.
Lemurs, lorises, and the T1.
Well, you missed one, the potos also. But lemurs, lorises, potos, and tarsiers. Those are the prosimians. Now, why are they not monophyletic?
We don't do the branch that goes up to the middle.
Yeah, so this would be their most recent common ancestor shared between all of the prosimians. But if we're just talking about the prosimians, that does not include all descendants of that most recent common ancestor, does it? That's paraphyletic. Yes?
How detailed he wants to know the phyletic trees, like plants and primates and all
You mean the phylogenetic trees?
that? Yeah.
Like, I'm not going to ask you to reproduce any of them, but I might give you one and then ask questions about it.
Okay.
Or I may give you pictures of a primate and ask you which major group of primates does this belong to. By major group, I would mean prosimian, New World monkey, Old World monkey, ape. And based on, I should be able to give you any picture of any type of primate, and you should be able to tell me that if you know the characteristics of each of the four groups. Even if it's one you've never seen before. I should be able to draw a made-up primate, and you should be able to tell me which one it is. Like, what are the general characteristics of the prosimians? Large eyes. What about body size? Pretty small. What about tails? Do they have them? Yes, they do. Are their tails prehensile? No, they're not. How about the, let's say the New World monkeys? What's that? Yeah, flat nose, prehensile tails. Larger body size than the prosimians, but still not particularly large. How do you tell the New World monkeys from the Old World monkeys? Mainly the nose. The older monkeys have that like downward facing nose. And then apes. That should be real easy. What do you see in apes? Not always. More erect, but not necessarily always erect.
No tail.
Generally no tail. That's a big one. Also generally much larger body size. Yes? Prehensile tail? Who wants to field that one? What's a prehensile tail? It can grab things. They can wrap it around stuff and actually hold on to it. So monkeys with prehensile tails can hang from a tree branch by their tail. Which one most closely resembles the ancestral primate? Prosimians. What was the ancestral primate like? How would you describe it?
Stereoscopic vision.
Yeah.
Tree grasping here. Climbing a tree.
If I were to ask you to describe its lifestyle, what would you say?
Lived in a tree named
It lived in a tree and ate insects. It's an arboreal insectivore. If I were to ask you roughly how long ago it existed, I probably wouldn't do that, but if I did, could you tell me? Yeah, about 65, 70 million years ago.
Do we need to know dates?
No, not really. I wouldn't ask you, you know, how long ago did the old world monkeys appear? I don't think that's particularly important. I might ask relative to the other groups, like if I were to say, which came first, the old world monkeys or the prosimians? You should know that. Or the new world monkeys or the apes? You should know that. Stuff like that. But I wouldn't ask you to give me years. Yes?
Is the main difference between hominid and hominid?
Yes. And general human characteristics. Hominids start to tend to look a lot more like people than any of the other apes. Yes. For what?
For taxis and kinesis.
Taxis?
Yeah, like stimulus.
Oh, yeah.
Yeah.
So behaviors, you're talking about taxis versus,
Yeah.
okay. So
Are you going to ask for like the specific names and types of that?
well, potentially, but it would only be down to the level of like phototaxis, chemotaxis, stuff like
And
that.
then the difference between kinesis and taxis.
Yeah. You should know the difference between kineses and taxis. Anybody know? Who can describe it?
Directed.
Taxis is directed behavior, kinesis is undirected behavior. Like an example of phototaxis would be you flip on a light and a moth starts banging into it because it's directing itself towards the light. An example of, I don't know, photokinesis would be you shine a light on some insect and it just starts skittering in every direction, every which way. All right, who has another question? Or I guess more specific to some of the stuff we were looking at this semester, chemotaxis would be those spiny lobsters moving towards conspecific aggregation cues. Chemokinesis would be some of the behavior they observed when exposing them to either conspecific or heterospecific alarm odors, where they started like, moving their antennas around and stuff like that. Not a directed motion. All right, who has more questions? Yes.
It asks what the derived characters of the embryophytes are, setting them apart from the green algae. What would those be?
So when we're talking about plants, what kind of characteristics do they have? So when we're talking about embryophytes, we mostly mean land plants.
Okay.
So what kind of characteristics do they have that the green algae do not? Yeah, alternation of generations maybe. What's something way more obvious? Yes, I mean stomata or cuticle or
Wait, aren't embryophytes like all?
yeah. So these may not necessarily be characteristics shared by all embryophytes, but there are characteristics that embryophytes have that the algae do not. So adaptations, basically all the adaptations that are specific to living on land. it doesn't necessarily, if I were to ask that question, it wouldn't necessarily have to be something shared by all plants. Yes.
How detailed do you want us to go with like the whole pollination for gymnosperm and angio?
So you should be able to tell me, now this is a tough one because I have to remember what I taught you guys and what I taught at my other school, and I went way more in depth on pollination there. So, how much did I tell you guys? Not a lot? Okay. But you know, you can identify all the parts of a flower, right? All the reproductive structures. So like if I were to give you a picture pointing to an anther or a, I don't know, stigma or ovary, you would be able to recognize those, right? You'd know which ones are male, which ones are female, which ones produce pollen versus eggs. You know where the pollen goes in on the female part of the plant. Some people saying yes, some people saying no. So the stigma, that's that sticky opening at the top of the lady parts of the plant. That's where the pollen goes in. Then it grows a pollen tube which travels down the style into the ovary. I will be consulting the lectures that I gave you guys while writing the test. So I shouldn't make any mistake in putting something on there that I didn't teach you guys, or that isn't in the lab manual. Basically nothing that wasn't in either the lab manual, my PowerPoint lectures, or the Isla Royale activity. Basically nothing that wasn't in one of those three things is going to show up on the test. I'll be reading those while I write my questions. Does that seem fair? Okay, good. Because that's how it's going to be one way or the other. Yes?
It says to like review all the slides and like print samples of you. How are you supposed to do that?
So in that case, when I say slides, I mean the lecture slides.
I thought you meant the ones we like made. You know? A little nervous.
I mean, the plant questions, I won't ask you stuff that isn't in like the lab manual or in the handouts you got to keep or in the PowerPoint lectures. You may be given pictures of plants and a dichotomous key, and you may be asked to ID what type of plant it was. Hopefully everyone remembers how to do that. I generally wouldn't pick something like the oak that had like 19 steps. It would be one of the simpler ones. I also wouldn't pick something as obvious as the orange tree. Maybe something you haven't even seen yet. But since you know how to use the key, you know how to use the key. If I do that, you would also be asked to list each step for the keys. That way, even if you make a wrong move somewhere, you'd still get partial credit up to the point where you got it wrong. That's good, right?
Yes.
Yes. All right, who's got more questions? We've only been here for an hour. Yes?
What do spores do in angiosperms?
So spores and angiosperms are just another, same thing they do in basically any other plant. They are used for reproduction. Yes?
I know, like with that question on the thing itself, it says understand the relationship between spores, pollen, gametes, seeds, flowers, and fruits in the angiosperm. Is that like how they all connect, or just like their personal?
No, I mean, I guess know roughly where they are in the life cycle, like which one gives rise to which one. So if I were gonna, like, what gives rise to pollen? It would be the gametified structures in the flower. And then, you know, pollen and ova would give rise to what when they fertilize? Seed. Seed. And then that seed would grow into sporified or gametified? Sporified, yes. I'll be careful not to be like unfair with those questions. Like I don't want you guys to fail. That makes me look bad too. At the same time, I also can't really write a test where everybody gets an A because that also makes me look bad. Unless you guys are all getting A's on the lecture exams too, and then it's fine.
I think it would make you look really good.
What would make me look?
You look better than
Yeah, you would look better than the lecture people, and then they'd be like, "Oh, you should teach lecture."
They would assume that my test was too easy if you guys score significantly higher on my test than you do on the lecture exams compared to your classmates. That's generally how they judge it. Well, I mean, also, don't worry about your grade too much. Worry about learning the material. That's what's important. If you learn all the material, you're gonna get an A. That's just how it works.
Sometimes it just doesn't. Like, that's the problem.
I mean, I guess if you're bombing the test intentionally, like not writing
No.
down the answers
Oh,
that you
no.
know. If you know all the answers, just write the right answers.
But then sometimes they just get you, and you're like, I don't get it. Like, I knew it.
If you don't understand the question, make sure you ask me for clarification.
Okay, see, that's where you went right. Sometimes people don't help us in lecture like you do.
So like, when you're taking my test, make sure you read the question completely and carefully before answering. And if you aren't 100% sure you know what I'm asking, ask me to clarify the question.
Thank you.
I absolutely am trying to test your knowledge of biology, not your ability to interpret my questions.
We need that. We need it.
Yes.
Boom slash
Yeah.
So what does it sound like? Like let's take it outside of biology. Let's say we're talking about the economy. If the economy's booming, what's going on? Yeah, everything's growing. If the economy's busting, what's going on? Everybody's eating. So you can apply that to biology as well. If you have a boom cycle in the ecosystem, or a boom cycle in one specific species reproduction, what do you see? Yeah, big numbers, big growth. If it's a bust cycle, it's the opposite. And to expand on that a little bit, understand how boom or bust cycles in one thing may affect related organisms. Like, if the mooses are booming, what's happening to the wolves? What's happening to the plants? Probably kind of crashing. Yeah.
Thank you.
Sure. Have a good night. Let me see if anyone's emailed questions yet. Although if they're free to email me questions, they should just be here. But I know a number of people have other tests right now, so they couldn't make it. Hence the video.
I think we should live stream the last 15 seconds of the rock.
That's 15 seconds.
Is the game on right now?
Yeah, there's
One time.
It's 1:10
Oh,
15 seconds.
okay.
What rockets?
Rockets are up?
Yeah.
Awesome.
I know. I don't know if it's boring. Oh, you have it on?
You already have it on?
Yeah.
Nice. I hate having to add. Oh, do I not have ad block installed on this? That's not a good idea. You can end up getting a virus on the school's computer. I bet it could be traced back to me. It should be basically instantaneous. Come on. Okay, maybe let's try a different one. Come on.
It's gonna be done by the time.
Yeah, it probably will be.
No. There shouldn't be those right now.
Oh, so the rockets are basically.
They still have my 5 seconds and they just somewhere.
Alright, there we go. Terrible quality, but it works. We'll get back to biology in just a couple minutes.
Oh, we're not able to do that. Don't look at the no reflection. Put it away. Oh, he's gonna make him better.
He better make it better. I was following the Blazer game last night. Oh, it's so bad.
Not in the mood.
See game 3? More overtime. I was so glad it came down to Jokic blowing that free throw too. I hate that guy.
Oh, no! Oh, that's not good.
Oh. All right, in the meantime, who's got biology questions? Yes.
What are the family and agios?
Okay, what is a gamete?
Uh,
In general, in biology, how would you define a gamete? More specifically, what type of reproduction? Yeah, so gametes are sex cells. They're eggs and sperm. So what do the eggs and sperm do in embryo? Same thing they do in basically anything else. Is that, did I say it?
it's not that big question.
Yeah, a haploid cell has how many copies of each chromosome? One. A diploid cell has. You should also know which structures are haploid or diploid in plants. Okay, sporophyte is haploid or diploid, and it produces spores through what type of division? Meiosis. Because the spores are haploid or diploid. And then they grow into the gametophyte, which is haploid, which produces egg and sperm through what type of substance? So it's
Oh my god.
oh no Curry. Oh! I love that they got two tries at it.
Two open threes.
Oh, they're two best 3-point shooters too. Well, yeah. You say it's better than Durant?
As a 3-point two main players.
Damn, they got two really good Lillard shooters in there.
I guess I saw them. Good stuff. So on that.
So in trees, now we're talking about how to gymnosperm drainage, right? Because there are no bryophyte trees. Now, what happens to the relationship between sporophyte and gametophyte as you go into the higher plants? Yeah, the sporophyte is dominant, and the gametophyte structure is reduced until you get to the point where, when we're talking about angiosperms, the gametophyte is microscopic. Yeah, it's tiny, tiny, and it's housed entirely within sporophyte tissue. So when it's in the lower plants, then you see larger gametophyte structures and less sporophyte dominance.
So really, the only ones that we actually know like what they look like in terms of like the actual plant are the ones that are like. So like,
Well, those are the ones where it's going to be harder to tell you. You should know what they look like in any of them,
yeah.
but it's going to be real obvious in the angiosperms. Watch me get this whole thing taken down from YouTube because there was a short clip of basketball in there.
Oh.
I may have to edit that out.
Wait, can you see the TV?
Yeah, I deliberately set it up so you can see the board.
I didn't even think about that.
But I can easily just edit that section out. if I have to. Although I'll be surprised if they actually find it before Wednesday,
Yeah.
and if they find it after that, maybe take it down. Who cares?
So hominoids
Yeah. Yeah. So hominoids is basically just another word for the apes,
Yeah.
which includes everything from the gibbons up to us. The hominids start out with like the Australopithecus. Those are the ones that look a lot like
Yeah, do you know that name?
these. No, I wouldn't ask you to reproduce that name, but if you see that name, you should recognize it as hominid.
So hominid is like, hominid is a hominid.
Yeah, hominids are within the hominoids, but it's more specific. Sort of like, all like a rat is a rodent, but not all rodents are rats.
For intertidal regions, when it says can you find data intertidal regions, just like high, middle, or low, like
Yeah, that's about as specific as I probably get.
so it'd be like maybe like a diagram.
Yeah, in that case, it'd probably be a picture. Yes.
For there's like the in-lab questions. It says plants evolve a cuticle. So they also evolve stomata, but what else is there like that evolved with the cuticle?
Well, what is the cuticle designed to do?
Keep in water. Less
So it keeps
acidification.
water from leaving the plant,
Right.
from transpiring out through the tissues.
Yeah.
What is it also going to prevent? Yeah, it's going to prevent water from coming in.
Right.
If water can't go through, can't go out or in.
Oh, so xylem.
Yeah. So then you will, not the phloem because that's not related to water.
Okay, so
but the
just-
xylem. So
okay.
then they would need. You could just say vascular tissue.
Okay.
So then, if the plant is covered with a cuticle, that means that the leaves, for instance, can't take up their own water necessarily. So you would need vascular tissue to deliver water to those tissues. So the evolution of the cuticle solved one problem and created several more.
Okay.
And then plants came up with further adaptations to solve the new problems that were created, like stomata and vascular tissue.
What kind of confused me in like the plant area is like one part it says what are the ancestral characters of all embryophytes, and then later says what are the derived characters of the embryophytes. So what's the difference between ancestral and derived? Because
So that's the question you're asking. What's an ancestral characteristic versus a derived characteristic? And we went over that more in the first half of the semester. So to say something is ancestral kind of implies that it was present in the ancestors.
But not present.
But, well, no, it will likely still be present unless it's been lost through evolution. But a derived characteristic is generally something that comes later. So like an ancestral characteristic of all of the, I don't know, hominids, let's say, that would include things like the presence of an opposable thumb or stereoscopic vision, because the ancestors of all hominids have that. Now, derived characteristics of the hominids would be things like bipedalism, increased leg length, increased height, increased brain size, stuff like that. Because those are things that are derived later on in the evolutionary history of the hominids. So ancestral characteristics came from the ancestors, possessed by all descendants unless they've been lost through evolution. Derived characteristics, not necessarily present in the ancestors, not necessarily present in all of the descendants.
okay, so like for example, an ancestral for the embryophyte would be like chlorophyll,
Yes.
and then like a derived would be
Yeah, or vascular tissue, or floppers.
Do you need to know
I don't think I'd ask too much about that.
There's only one thing on that, it's like the difference between the phaylum,
Which were?
afarensis, habilis, and brachis.
So, what's one thing that is characteristic of Homo habilis that was not characteristic of Australopithecus afarensis?
Tools?
Yes, tools. And what was the next one? Erectus. What's fire,
And
or more upright posture, or taller. So, yes.
Do we have to know how to calculate p-value?
I don't think I'd make you do that by hand. This isn't a stats class. But you should know what it is, be able to define what it means, and be able to interpret it if given p-value. You don't have to calculate.
What does significance mean in that?
So significance means that you can confidently say that the differences between two groups are because those two groups are fundamentally separate. Like, you could say, let me come up with a good simple example here. Like, for instance, if you have two groups of people and you're looking at height, if the differences in height between those two groups are significant, then you can say that they are two separate groups of people, not just two samples of the same population.
What is
Like,
that?
like you could say that maybe one of them is men and the other one is women, or one of them is adults and the other one is children,
Oh,
or
comparing it
one
to
of them is
actually
Dutch
known
and the
value-
other one is Chinese.
To actual known values? Is that what it means?
No, it means that you can confidently say they're separate populations. That's
Not random.
yeah, that they're not just two random samples of the same population.
That's when it's less than 5.
Okay.
Is it always a 0.05?
No, that's called the alpha value, and the alpha value is determined by the researcher.
Okay, so we will be given that.
But if not, assume 0.05. 0.05 is the most common alpha value. So unless someone says otherwise, like unless someone specifically says alpha value is set to 0.1 or 0.01, you can assume 0.05
On the module,
for the purposes of this class at least.
on the module we did for the last lab, it said like for like it's like a more narrow value or something.
Oh yeah, there's like narrow.
Yeah.
Something like that.
I don't
I felt like it wasn't very important.
like
And-
for saying it was like the
Is it talking about the
p-value-
t statistic?
I think it was about p-value because it was saying like if it's significant then you're looking at it or like the narrower like the range like the more
That sounds
significant
I
it
think
is.
it might have been like for growth, for like long season versus.
I'm not quite sure what you guys are asking.
I know what you're talking about. I don't know.
I wish I remembered.
Well, if you come up with it, send me an
Yeah,
email or
I
something.
will. What are body plans?
So body plan is like the basic layout of an organism.
Okay.
So like the human body plan is a bipedal tetrapod. We're bilaterally symmetrical. We are cephalized, meaning we have a head.
Okay.
As opposed to something like a sea star, the body plan is, pentaradially symmetrical,
Okay.
something like that.
But green algae isn't divided into a body plan. Because that's what I put on my answer, that the difference between bryophytes and green algae is that bryophytes have a body plan and that
That's not necessarily true, because something like kelp is green algae, and that definitely has a more or less defined body plan.
okay.
Plants, any kind of plant in general is usually going to have a less defined body plan than animals.
Okay.
Like you look at a human, you're always going to see, or you're almost always going to see 2 arms and 2 legs, and you're basically never going to see more than that.
So
With a plant, like with a pine tree or something, you might see one with 12 branches, you might see one with 112 branches.
okay.
So the body plan is a little bit less definite in that case.
So then the difference between bryophytes and green algae would be the alternation of generations.
That could be one. Let me see if I can think of a better answer for that. I think that's the best one I can come up with.
Yeah. Okay.
Because the bryophytes generally don't have the any of the more derived characteristics of embryophytes.
Right. Okay.
I doubt you would encounter that question on my exam.
Okay.
I try to make the questions like pretty obvious this or that kind of thing. I don't like giving trick questions. The closest I'll give you to a trick question is something that's obvious to a person that's studied, but completely tricky to someone who hasn't.
Right. Okay.
I feel like the most confusing thing to me, at least after I applied everything else, is like the primate section. We didn't really go over that in lecture that much.
Right.
So like, okay, so primates
I'll
have,
try to keep that in mind while writing the test as well.
Yeah. So they have like 4 main, like on a, like for the phylogenetic tree, there's like 4 main sections, right?
Yeah.
It's like the
Prosimians?
simians and then the hominoids and the hominoids. Or is there one in the middle?
There's 2 of them.
Oh,
So
New
yeah,
World.
prosimians, New World monkeys, Old World monkeys, apes.
That's kind of confusing to me because New World is before, right? New World came
Yeah.
before.
Right, but remember what that actually what's the New World? This is very Eurocentric and a really dumb way of classifying things because like we act like the New World was discovered later, but it was only that Europeans became aware of it.
Yeah.
It's kind of racist. Yes,
For like a lot of the primate questions for
but we
like-
still use it because it's an easy designation.
Yeah.
All right, sorry.
No, don't worry about it. For the in-lab questions, I feel like a lot of my answers were relating back to like the most recent common ancestor. Like there's this one question about how canines vary across group, and then like what might that tell us? So like
How
canines,
canines specifically vary across group?
yeah, vary
Okay.
across the group. It says I said some have like very large and prominent canine teeth, and then some don't, and that tells us that the MRCA of hominids had large canines for meat eating and maybe grabbing. Like,
So it doesn't necessarily tell us that. I mean,
Right.
that's probably the case, but it's not always going to be the
Right.
case. So large canines in primates, do we have particularly large canines compared to other primates? No. What is one thing you might think that those other primates could use those large canines for?
Crushing things.
Eating, maybe.
right.
What might be another thing they might use them for? Why wouldn't we need them?
But we don't have to crush them all.
Well, I mean, but if we do want to crush their bones, how do we do it?
Yeah, like
A tool. Yeah, exactly. We would use a tool. We use a bone cracker. Because we can. Because we're smart enough to make them. Maybe they might also be used for fighting. Essentially. Why wouldn't we use our teeth for fighting? Yeah, because we've got swords and guns, stuff like that.
So to like answer this question, we're saying like the advantages and disadvantages of having like large canines in the lab.
Well, it's not necessarily advantages versus disadvantages. It's the fact that we don't necessarily need to use our teeth for things that other hominoids might,
Okay.
because we have better tools.
And we can.
So our teeth become much less of a tool, much less of a weapon, and much more specifically adapted just to.
Do we need no specifics about teeth? Because like, it doesn't say anything in here. I know we went over it a little bit.
I mean, you should know generally what type of foods each type of tooth is good for. Like molars, for instance. What do we use molars for? Yeah. Grinding. Yeah. So if we're eating plants or something like that, the molars become useful. That's why you don't see molars in a cat, because cats don't eat plants. All they eat is meat. So all they need is to be able to tear little pieces off and then swallow.
And I think canines are more for crushing.
Not so much for crushing. Canines are more for like biting into something and holding onto it, and for like ripping pieces off. And incisors are for, taking bites out of stuff. So we have very diverse teeth, as do all of the hominoids, all the apes, because we have a pretty diverse diet.
Yeah, I started reading that like as we started to progress like farther into the news, like hominids diet is like
The more advanced they become, the more general their diet becomes, because we get to be better generalists. We get to be good at harvesting plants, but also good at hunting animals, things like that. Yeah, we get smarter. And when we start living in communities and using tools, that opens up a whole new source of food because now we can take down elephants. You're never going to see a gorilla eat an elephant, but early people did, and some people still do.
There's this other question. It says, what might we conclude about the feeding habits of the gorilla from the skull inspection compared to other hominid plants?
You know, I probably wouldn't put that on the test because I don't really want to have the skulls out because we only have a couple of them. So, yeah, I wouldn't worry too much about it.
Because like, it's just like a lot of these questions we're asking for like, what can like we assume if like they have bigger teeth or like a larger skull or like.
Yeah,
Like
and
that
you might
kind of
get something
stuff.
like that. Like if I were to say, if you observe the skull of some unknown ape
Right.
and you see large, prominent molars, what could you conclude about something about the diet of this organism?
Oh, okay. So it's more of like a specific question on.
It could be specific or it could be a little bit more general, but I'm not going to have you looking at the actual skulls. I might just describe something to you and say, what inferences can you make from that?
How detailed should that be?
Well, I mean, for that example specifically, if I were to give you exactly that question,
Yeah.
something with large, prominent molars likely has a diet that consists at least partially of plants.
Right.
Or you could say that plants are an important part of their diet.
There's molars, canines,
Incisors and
other premolars. And that's like in the book.
yeah, some important molars. Yes. I got a question. You stretch.
What are incisors important for?
So incisors are used for taking bites out of things.
Okay.
Like if I give you an apple, you're gonna get pieces off of it using your incisors. Or if I give you a cheeseburger, you're gonna get pieces off of that using your incisors.
Thank you.
Sure.
See you guys.
How long is the?
I usually aim for having the vast majority of the class finish in between 45 minutes and an hour and 15 minutes. I'll probably set a 2-hour time limit on it, and I will aim for the vast majority of it being done way before that. Like, if you think about the last one, that one was basically perfect. I don't think anyone was done in less than 45 minutes, but most of the class was done in less than an hour and 15 minutes. If someone comes in really knowing their stuff, they'll probably be able to bang it out in half an hour. But I'll be impressed with that.
I feel like the most confusing thing that someone asked was the facial projected.
Yeah, I mean, there's not going to be a whole lot of detailed questions. Just understand the general trends. Like, what happens to the prominence of the brow as we go from early hominids to Homo sapiens? Gets reduced. Stuff like that. If you understand the general trends, that'll be good. And I won't get super species specific. Like, you'd never see something like compare the facial projection index of Australopithecus africanus and afarensis. If this were an anthropology class, you might see something like that, but it's not.
Alright, thank you.
All right.
One last thing I already asked it though, about the basic animals at the time tools.
Yeah.
So I should know like kind of how they look, like picture.
You should know what a chitin looks like. You should know what a mussel looks like. You should know what a crab looks like. Hopefully you already know what a crab looks like.
It's like chitin, CHI2.
Yes. So a chitin is a mollusk, and it's Chiton, not Tien. Chitin is a biological material used in like fungal cell walls and crab shells. A chiton is one of these guys. It's a polyplacophoric mollusk.
It kind of looks like a mussel.
It doesn't look anything like a mussel. You kidding me?
Like the rounded part.
It has an 8-plated shell on one surface, and it has the foot on the bottom, and then the mantle extends beyond the shell. That looks nothing like a mussel. You kidding me?
Like the rounded part looks like a mussel.
All right, I'm gonna.
Yeah.
Looks nothing like that.
You see it?
Not at all. So this is all shell, and it's shell on both sides.
Yeah, but like that round part looks the same.
It doesn't look the same at all. They couldn't be much more different. Like, look here, you have 8 plates of shell, and then this stuff right here is not covered by shell. That's mantle. Looks totally different. However, they are both in the same phylum.
Yeah.
They're both mollusks. But this is a polyplacophorid, and this is a bivalve.
Okay.
They are very different and have very different lifestyles. Like, for instance, these guys are sessile as adults. They anchor to a rock and they never leave. These guys are modal as adults. They can move around.
I put way too much detail into this. Like I put.
Also, some of them look really wild.
So you're just gonna like show us that picture of one of these?
Yeah, you literally might see this picture, and I might ask you, what?
Let's hope it's just the two of us.
I might ask you, what organism is this? What phylum does it belong to? Something like that.
Okay. So like, I put like a bunch of stuff. I put like
Strictly
crustacea.
strictly the ones that we saw.
Yeah, I'm not going to pull out anything that wasn't there. And in fact, I'm probably going to aim for the easier ones. Like I said, like I don't want you guys to fail this test.
Yeah. So like a chitin's a bivalve, so we know that.
The chitin is not a bivalve.
I mean,
Mussel is a bivalve.
oh, okay, the muscle's a bivalve.
It might be worthwhile knowing that, but I wouldn't ask that specific of information on a lot of these. Like, I'm not going to expect people to know that this is a polyplacophorid because we didn't go over it.
Yeah. Because I put all that detail into here. I don't think I needed it.
So, like, bivalve is the class, right?
Yes.
It's almost certain that I wouldn't ask you to give me the class. No phylum and no general type of organism like chitin or crab or limpet or something like that.
Yeah, mine. I don't know. So we don't need to know the specific.
You might need to know the sea hair.
Yeah, he said.
You guys should be able to recognize that guy.
That's a sea hair.
Yeah, we all saw those on the tadpole trip.
I remember like seeing.
There's also been a bunch of them living in that tank for the last couple of weeks, but I think they all died.
What is it?
So a limpet,
What's the secret on
those are the ones that look like this. They look sort of like a little volcano.
See, that kind of looks like the.
This is more similar to the kite than the bivalve.
Yeah.
Your toothfish.
What's a limp? What is it on
What would you guess? What phylum would you guess this is?
It looks like.
It is mollusk.
And then the sea hare was.
Sea hare is also a mollusk. A lot of them are mollusks. Let's look at one that's not a mollusk.
Toothfish.
That's. I think that's pretty simple.
So these guys are not mollusks.
That was a hard one.
These are arthropods. Gooseneck barnacle.
Any sea star is it?
Echinoderm.
I can't say it.
Actually, you know what? Let me double check on what phylum the sea hare is in. The sea hare. I'm second guessing myself.
It says mollusca.
Okay, that's what I thought. gastropod mollusks. I was worried that it might actually be an echinoderm because these are echinoderms. Oh, that's a terribly small picture. That's better. That's an echinoderm. The sea cucumber. But we didn't see those, so I'm not going to put that picture on the exam. I would like you to know it, but I don't think I could expect everyone to know it because I don't think everybody saw it. But they're easy to tell because so the woolly sculpin is a small fish, looks like that. The opali is really easy to identify because they have a white spot on the dorsal surface on either side of the dorsal fin that looks like two eyes looking up, and they're opal color.
So the quinotic, because those are both quinotic, right?
Yes.
So they most of the time quinotic kind of like fish or are fish.
Oh, no way. I mean, in the intertidal zone, yes.
Yeah.
But you're also a chordate.
Yeah. Well, okay.
Usually that.
Yeah, I think the only chordates you would see in the intertidal zone, at least marine chordates, would be fishes.
What was the animal that looked like honeycombs? Because I have like a big name for it, but I don't know. I look for it.
Honeycomb worm?
I just don't know.
I said it's a tube. I think tube fish.
It's not a tube fish.
What is a tube?
I'm not sure I've ever heard of tube fish. Let's
Alright.
Google that and see what let's see what comes up.
Well,
Okay.
you
yeah,
saw
that-
one of those?
No, we didn't see that. That will not be on the test.
It's the fresh tuna.
Yeah, the cow.
Yeah.
No, mine was.
That sounds like the honeycomb one. Not a fish. It's not a vertebrate.
It looks like a flower.
Kind of, but what is it? What's it called? It's one that most people always mispronounce. The what? Anemone. Anemone. Okay, most of the things that came up are flowers when I Googled that. But this is an anemone. Or this. Hopefully, yeah.
I want to, but we're just near him.
If I'm not too busy writing this test.
Yeah, I think I need to study more.
Probably. Where from?
I was born in Southeast, and then I went to high school outside of Portland. How well do you know the area?
I'm from Northeast Portland.
Okay, so I went to Westland High School.
Oh, you went to West
Yeah.
End.
But down here, if I tell people I'm from Westland, they're like, what? Where? So I just say Portland.
I went to Central County.
Oh, cool. You would not know any of my friends from Westland. they probably graduated high school right around the time you were born.
Born. Where in Southeast Florida?
I was born in Portland Adventist Hospital.
Like my dad's whole family, like my whole dad's side of the family is smart.
Okay. That's a good part of
Yeah,
Portland. That's been nice for a long time too. Like a lot of Southeast is getting nice now.
Yeah.
Because like the
I-
hipsters flooded into Selwood and gentrified the hell out of it.
Yeah.
But Eastmoreland's been nice for a while.
And then like the rest of it.
Yeah, none of my family's actually from Portland. Like my dad's from Texas, my mom's from Wisconsin. We moved to Portland in like 1980.
Dude, they really need to win a lot.
They really do. If we go down 3-2,
It's gonna be around.
I think they'll win it again in Portland. It's just like it's scary to go game 7 and
yeah. And I don't know if we're gonna be able to take 2 in a row in this series.
Yeah.
It's
tough series.
I guess we're gonna have to take 2 in a row. But, yeah.
Either way.
Well, no. Yeah, we will have to stake 2 in a row.
Yeah.
Yeah.
One of the teams is gonna have to.
Well, I guess not. If we win 5 and 7, we don't have to stake 2 in a row.
Yeah. That's both ways.
That's both in Denver, which I don't see happening.
Not a good. Dude, I think Dame will be mad because he didn't play amazing yesterday.
No, he didn't.
He was making like that last play when there was like not the last play, but we were down by 2. There was like 14 seconds after each drove, and then missed.
Somebody needs to start talking some trash. Like I need
Yeah.
Jamal Murray to start talking about how he's like beating up on
Yeah.
That'll
Because that would get Dame going.
Yeah.
But that's the problem. You don't want him to be like, oh, I'm only gonna go off if someone starts talking.
I
smack
know,
on
but he
me."
does. Like I need like Charles Barkley to start talking about how Dame's overrated or something.
Charles Barkley
Charles
said
Barkley
that they're going to the
thinks they're
final.
gonna
I know.
Finals.
Charles Barkley loves them. Which I mean, I could see us going to the conference finals,
Yeah.
but I don't know if anybody's being hosted.
If it's, that's what I was saying. If the Warriors win, no one's going. But if the Rockets win and the Blazers win, there's a chance.
Yeah. If the Rockets win, either the Nuggets or the Blazers could
Yeah.
potentially beat the Rockets.
I don't think the Rockets are good enough just because of the way it's just Harden. Like there's no offense.
I mean they're good enough to win it, but they're not